master mix aliquoted into a PCR strip tube and our single cell suspension sitting on ice.
Now we are ready to combine our master mix with water and our single cell suspension. The optimal cell loading concentration for this protocol is approximately 100 to 600 cells per microliter. Ensure that the cell suspension is diluted to the optimal cell loading concentration. See the user guide for details. We will refer to the cell suspension volume calculator table in the user guide. The left column lists the cell stock concentration in cells per microliter. The row at the top lists the targeted cell recovery. In this experiment, we are targeting 500 cells, and our cell suspension contains 500 cells per microliter. As a result, we need to load 40 microliters of water and 3.3 microliters of cell suspension into the master mix. Never mix cells directly with water. First, add water to the master mix, then add cells to the master mix containing the water. Add 40 microliters of water into the aliquoted master mix for all 4 samples. The master mix should be on a chilled ice block.
Now we are ready to add the single cell suspension into our master mix.
Gently pipette mix the single cell suspension just before adding the appropriate volume into the master mix.
Depending on the cell type, the cells may settle down to the bottom of the tube.
Therefore, when transferring the cells into the master mix, hold the pipette toward the center of the tube and dispense them directly into the master mix containing the water.
We have combined our master mix, water, and cells. Now we're ready to load the Chromium Next Gen chip.